Our first speaker today is Marissa Saunders. Marissa is Senior Director, Data Science at Recursion Pharmaceuticals. Marissa has oriented her career around using AI and machine learning to answer impactful questions, using technology to turn data into insights. At Recursion, she is leading efforts at the interface between deep learning and models to accelerate discovery and decision-making to enable drug discovery at scale using automated omics experiments, AI and machine learning predictions, and analysis and standardized workflows and decision engines. Prior to Recursion, Marissa worked in the learning space using language models to develop a taxonomy of skills, and as a big data scientist consultant working in fields ranging from biotech to video games. She has received a PhD in chemistry from University of Chicago and has completed a postdoc at University of Utah in structural biology lab, turning cryo-E electron microscopy images into protein structures. Marissa, back to you, I think.
Thank you so much.
You have 25 minutes for the talk, and I'll give you a notice at around 22 minutes, if that's okay.
Perfect. That sounds great. Is everyone seeing my slide okay?
Yes.
Perfect.
Looks good.
Wonderful. Well, I'm so happy to be here. Thank you so much for the invitation to talk. I am Marissa Saunders, and I am excited to share today's how we're thinking about industrializing drug discovery using in silico models in data loops with physical experiments. To orient you, if you aren't familiar with Recursion, we are a tech bio company with our headquarters in Salt Lake City, Utah. We also have offices in Milpitas, Toronto, Montreal, and London. So you may be asking, what is this tech bio thing? I thought there was a term of biotech. Well, what we think at Recursion is that many biotech companies have built these point solutions. So they have a particular tool, process, model, or analysis that they use to accomplish an important step in drug discovery. And this is where we started too. We started approaching discovery and screening using morphological profiling and specifically using cell painting. But as we continued on this journey, we realized discovering and developing medicines requires hundreds of steps. And if you really want to accelerate getting medicines into the hands of patients, you have to industrialize drug discovery. And that means you have to take these point solutions and integrate them as modules across many diverse steps. And so this is the pipeline that we use to think about how we move a drug from hypothesis all the way through the clinic. And you can see that there are many different modules along the way. So this idea of using point solutions in many places across steps is the first of the key ideas that I hope that you will take away today. The other two, which I'll introduce here and we'll talk about through the course of this talk, are that the most efficient experiment one can run is no experiment at all. And so that is the real promise of in silico methods. They help us to predict which experiments are the right experiments to run. And that means that we can be more efficient in both time and money in getting through drug discovery. To be able to get better and better at knowing which experiments we should run, we need better models. And for that, we need better data. And so a key idea at Recursion is that in order to have these virtuous cycles of learning and continue to improving our models, we have to couple the physical experiments that we use to progress programs through with in silico methods so that we can iteratively improve our models. We start by thinking about what are the data modules, where are the places that we need to focus. And you can see in this pipeline, we have a range of different data points where we're collecting data, ranging from patient data that help us build causal ML models to target predictions to our scaled omics capabilities. And all of these different data modalities then help us to navigate through drug discovery. We use ML models and AI models throughout this process to help inform which experiments we should run. So in the patient connectivity and novelty stage, as an example, we use LLMs to filter through the corpus of data in the world and help us know where we should be interested in looking for new hypotheses. When we have patient data, we use causal genetic models to help inform what are unknown targets or causal genes for drug discovery. When we move to hit and target validation, we use generative chemistry models, which you've heard about earlier yesterday, in order to generate the structures that we need to be feeding into the system. And we use foundation models for imaging, which I'll talk about in just a minute, in order to interpret our scaled phenomics data. When we go into compound optimization, we're using protein ligand binding models to identify what is the actual protein target that compounds are binding to. And we use chemical property prediction in order to know which things are actually going to move us to a good space in terms of ADMET properties. And then finally, in translation, we use things like video analysis models and active learning models in order to tell us which compounds are starting to show effects of toxicity in our mice, and to better truncate those experiments if we need to. And so in order to build all of these models that help us to filter, we really do need large amounts of data. And to get there, we lean into this flywheel idea where we have real physical experiments happening in high-dimensional assays at scale. And this data then feeds into digital representations of reality. So we generate this corpus of data. And then that data in turn is fed into our algorithms and our methods in Silico in order to help us create these in silico models that inform in turn which experiments we should run next. To do this, what we're doing is we're really using this drug discovery process to generate a data set that is fit for purpose for new ML models. And when we do this, we think about the data a few characteristics. The first characteristics is that these should be universal or near universal assays. So we don't want to be running assays in one disease program that we can't apply in another. And the other characteristic is that we would like this to be high dimensional data because there is rich signal in these high dimensional interactions. And so as a result, the kinds of data that we collect at scale are omics data, genomics, transcriptomics, proteomics, and phenomics, in addition to some standard ADME assays, and then some mouse models and real-world patient data. And over the years, we've managed to collect a significant corpus of data across all of these layers, either through partnerships or from generating that data in-house on our platform. And you can see our phenomics layer is our most prolific. It is also the most cost-efficient assay in our stack. And you can also see that there are several areas, real-world patient data and genomics data, where we partner with other people in order to have access to some of this data. And then there are also layers where we are just now starting to enter this flywheel. Proteomics, we're just starting this, we started earlier this year, and our ADME platform, we've started running at scale late last year. What all this data gives us is the ability to build a range of different models. And you can see here, we build everything from causal genetic models through to chemical property prediction models, and even video time-lapse behavioral models for our in vivo mix programs. I'm going to start by talking about phenomics first, because as I said, this is sort of our foundational layer. It is the place that we often start in thinking about programs. The idea behind phenomics is that morphology is a distal endpoint to capture holistic cellular response. And so if you think about perturbing a cell by applying some sort of disease perturbant, that perturbation is going to change the gene expression, and so change the transcription activities in the cell. It's also going to shift protein levels as a result of those changes in transcription. And that in turn is going to shift protein activity. And all of these things are things that we can measure through different omics technologies, transcriptomics, proteomics, and metabolomics. But phenomics spans all of these. The morphology or the endpoint of this cell is informed by all of these changes, resulting in a cellular end state. And we in fact see if we apply different perturbations to cells that we can visually distinguish between some very different morphologies. And so this is the foundation on which we're building. When we started with this model that what we should do is take cells, apply some sort of a perturbation to get them into a disease state, and then screen all of our compounds to see which of those compounds rescue that disease state back to a more healthy state. And so this worked. We drove several programs using this methodology. And what was exciting about this was that we could test a range of different perturbations. We could do CRISPR knockout, we could add secreted factors, we could physically stress these cells out, and all of those things could be captured on our platform. But in this modality, we were still in this place where for every different disease that we wanted to test, we had to screen all of our compounds. And so what that meant is if we want to think broadly, if we want to screen across a wide breadth of diseases, we have this quadratic scaling where we have to screen the entire library for each one of those. And that was not something that would scale the way that we had ambitions to scale. And so the way that we solved this problem was to think about using an inferential map. So the idea was, instead of this paradigm where you have a disease and on top of that disease you put a compound, the question was, can we take a disease and then separately take a compound and treat cells with those, and in some way from the images capture biological relationships in such a way that those representations let us infer something about what will happen if those things are put together. And so what we've come up with is these phenomaps. You'll see this picture in a few slides here. The red squares are where those two things are very similar to one another, so the images look similar biologically, and the blue squares is where they look opposite from one another in this biological representational space. Now you might say the key thing here is then to figure out how to transform an image into a representation that carries this biological meaning. And how do we do this? The way that we are doing this, in fact, is through deep learning models, and most recently it is through foundation models. And these models enable us to take advantage of this flywheel of data generation, as I'll explain now. So we have generated to date over 250 million different experiments ranging across 50 human cell types, including 1 trillion different human neurons that have been generated. And what we can do with all of this data is we can feed it into a foundation model, which is a model where we're not telling the machine what the answer is that we want in terms of a class, but we're rather asking it to do a task. And the task that we're asking it to do is to reconstruct a masked image. So we can take this image, which is our input image on the right, and we can mask out a large chunk of it, say 75% of the image gets masked out. And then we ask our ML algorithm to reconstruct that image from this masked input. And as you can see, it does a very, very good job of doing that. And what's more important is this task captures information about the biology that's going on, about the morphology of these cells, that then we can use those embeddings to represent our perturbations in this inferential map. One of the things that's really exciting about this model is that we are seeing that it continues to scale with the number of parameters. So we can build larger and larger network models as we get more data and as we have access to more compute. And that means these models will just get better and better. And that's really desirable. We've also seen that these models are generalizable to new problems. And so if we compare this to other ways of embedding image data, we see that it does significantly better at recall and actually is able to capture things beyond just the context where it was trained, which is the great promise of foundation models. Phenom Beta, which is available through NVIDIA's BioNeMo platform for anyone to use, is one of those models, and we continue to iterate on this internally as we generate more and more data in this flywheel of improvement. And so when we do this, when we put together this map, what we can do is we can look at the map and we can say, for any combination that we want to think of. What is the signal that we're seeing in the map? Are these things similar? Do they have no relationships, or are they opposite to each other? And we can do this not only for gene-gene relationships, where we know these signaling pathways and we see that the map recapitulates these signaling pathways. We can also do this in comparing gene to compounds. And so we can see for some known inhibitors that the maps are also recapitulating the biology that we know from literature and from prior experiments we should see. And then we can combine those into one so that we see both the compounds and the related genes and get a full picture of the known and unknown biology around a particular area. Once we've got the omics data, we want to also have a method to validate that, or to have a orthogonal assay to show us that what we're seeing is not just an artifact of how we're doing phenomics, but it is actually real biology. And the way that we're doing that is through transcriptomics. This is our second data layer. We have generated at scale the first genome scale transcriptomic map through Perturb-seq. And what this does is it not only gives us a way to connect between genetic knockout and transcriptomic signature, which lets us tie it back to patients, it also has given us this multimodal workflow in transcriptomics and phenomics that is able to predict which compounds will fail downstream tests and which compounds will pass downstream tests. And that lets us run the right experiments at the right times. Other models that we're using right now, we're using patient data to create causal models. So in this model setting, the data that we're using is partnership data from Tempus and Helix, which have patient records that are de-identified and standardized. And by combining this data, which really does forward genetics, where we know the phenotype of the patient and we can map forward what are the genetic drivers that are associated with that phenotype, we can couple that with Recursion's platform, which lets us knock out particular genes and observe a phenotype in cells. And so we get this virtuous cycle of learning where we're using both forward genetics and reverse genetics to get to novel targets. Once we have those targets and we have compounds that look like they're affecting them in phenomic space, we can also then add in silico chemistry models like our matchmaker model that predicts which proteins a particular compound will bind with. And so from this model, we actually have over 2 quadrillion different protein ligand interactions where we've scored how likely it is that those two are interacting. And that lets us go from this phenomics target, which is a little bit more fuzzy than an actual binding target, to being able to hypothesize what binding targets might be. And so does this work? Does this actually make a difference in how fast we're able to move or how many different hypotheses we're able to test? So we have data on the first 2 stages here of a volume that we can start to aggregate. And what we see is before we did this industrialization process in 2022, it took a team of 40 people and we were able to explore 30 program hypotheses. The following year, when we had started this industrialized process, that team was down to 7 people, and the number of program hypotheses had exploded to about 115 over the course of the year. And then if we look at the first half of 2024, this year, you need less than 1 full-time equivalent person to get programs from initiation all the way to where we're nominating them for hit to lead. And we've been able to do that for 201 program hypotheses going into this system. And so what that is really doing is it's enabling us to shift the timelines and the shape of the drug discovery funnel from a very V-shaped funnel that takes 1 to 2 years for this early hit discovery and millions of dollars to something that takes weeks or months and can be measured in the thousands of dollars. So in the time that I have left, I would like to talk through a case study to show you how this might be applied to a particular discovery program. I'm going to talk about RBM39. This is a program that was one of our earlier programs, and so it doesn't have all of the modeling that I've talked through, but it is one that we've brought all the way through to clearing IND, and so I think it's a great story to tell. So CDK12 was initially identified as a target for HR proficient cancers, but the challenge is because this is a kinase, we had non-specificity issues in finding an inhibitor for CDK12. CDK12 and CDK13 are very similar, and so most CDK12 inhibitors ended up also inhibiting CDK13, and that caused a lot of toxicity. When we looked in our phenomap at CDK12, what we saw was there's this gene, RBM39, where the knockout of RBM39 mimics the knockout of CDK12, but it doesn't show that same relationship with CDK13. And so this seemed like a great target for getting the effects of CDK12 without having the inhibition of CDK13. When we screened small molecules, what we saw was that there are small molecules that target RBM39 and show similarity to CDK12 without having any similarity to CDK13. And so this felt really, really exciting. With our matchmaker product, which predicts the binding of compounds to protein pockets, we were able to identify, or we were able to predict that these new compounds were not in fact CDK inhibitors. And so here 2 is a significant similarity score. We can see that positive controls, CDK12 and CDK13 inhibitors are scoring as being predicted as being high binders to the kinases here. But these 2 new molecules, these recursion internal molecules, are not predicted that way. We can physically screen to confirm that digital hypothesis and run an experiment that matters. And what we can see here is when we screen for CDK12 activity, we can see that CDK12 inhibitors are actually binding to CDK12. Our hit compounds are not binding to CDK12 despite having the same phenomic effect. And so that led us to feel like this is a really exciting direction to take this program. In order to develop that compound and get it to a compound that we can dose in an animal, we have to iterate on the compound design, and the phenomics map actually gives us a way to test that concentration response and to test it not just against CDK12 and RBM39, but also against CDK13 to make sure we're not picking up activity that we don't want. And so here you can see as we increase the concentration, we can evaluate similarity in the map to these different targets and get a nice concentration response curve that allows us to iterate on improving molecules. And that lets us drive structure activity relationships. We can see here that from this compound, we're able to make variations, change some of the R groups, and then profile within the phenomic map to make sure that we're maintaining the response that we want to see. And when we test for RBM39 degradation, we see an echoing of that effect. And so we know that the phenomic inference map is telling us what the physical experiments would tell us as well. And then finally, excitingly, this compound got to the place where we were able to dose it in a tumor model. And so what we see here is with our hit compound, we see a significant reduction in the tumor volume over time. And we also see an increase in our survival rates over time compared to the vehicle. And so what this story should illustrate is that these predictions and minimal standard experiments will let us rapidly and efficiently identify a development candidate. These are the numbers for the RBM39 program, where you can see in silico, we started very, very wide. We were able to screen a very large number of molecules in silico. We were able to, in phenomics experiments, go through a smaller set of those compounds and drive relatively quickly to candidate quality compounds. And we were able to do this in a relatively short time. It took us 9 months from starting this program to having something that felt like it had an appropriate therapeutic index to move forward with. And in fact, this compound just cleared IND earlier this month. And so we anticipate starting phase 1, phase 2 trials for that at the beginning of next year. And so in summary, we're really excited about this idea of data loops, this idea of running experiments where we need to in order to improve our models, which will tell us better which experiments we need to run. And the end game of all of this is that patients are waiting for treatments, and we have to find new ways to accelerate this. And in silico models, such as the ones we've talked about so far today at this workshop, are the way to move this forward. That's all I have.
Thank you. Thank you so much, Marissa, for an excellent presentation, actually. I'll see if there are any questions. There are some questions in the chat, which I will read. Marissa, if you can answer those, that will be great.
Fantastic.
I'm excited to learn more about the matchmaker model. I'm curious what inputs were used for the model. Was it trained on different protein ligand complexes, or were the proteins and ligands provided separately?
So it was actually trained on a large suite of data, including known protein ligand interactions, including biochemical assay information for binding. And so that was the input, and then it has been shown to be able to make these predictions for things that it had not seen before. So it does rest on that kind of data.
We'll take one more question. It's great that the model helps a lot in time and cost in early lead discovery. What's your strategy to do the same in the clinical outcome?
You know, it's a really, really good question. In the clinical space, we know that early clinical screens are relatively small in terms of patient numbers. We run the first few cycles through in a small handful of patients. And so it's really important to be able to get the patient profile right, because we know that in this age of personalized medicine, particularly in oncology, each patient is slightly different, and what we want to do is identify the patient cohort that best fits our treatment. And so one of the ways that models can help is in finding the right patient population to bring into our clinical trials. There's also more logistical things that we can do in the clinical trial space in terms of knowing how best to recruit people into these trials in order to identify what is the population that we need to open this up to in order to get enough people enrolled. And so I think there are a wide range of ways that we can use some of these same types of strategies in order to accelerate clinical phases.
One more question. Can you please shed more light on, on validation of model, and in case of isoforms, how selectivity and specificity were handled?
Yeah, so I'm happy to actually refer you to some of the publications around Matchmaker. I don't have those citations on hand. I'll look them up offline here, and I'm happy to send that on to you. In general, most of the work that we're doing here at Recursion, most of the papers that we've published are available on the Recursion website, so recursionpharma.com, and there is a literature kind of an area there that you can look at and see some of the backing science for these models.
One last question I'll go with. How to elucidate drug mechanism of action based on phenomics data?
You know, that's an awesome question, and it's something that we think about a lot. I think the term mechanism of action is actually a little bit fuzzy, and it means different things to different people. So one of the things that we think about when we think about mechanism of action is what are the general biological pathways that are being activated? And to answer that question, we use the phenomics data and the transcriptomics data, and we're able to find associations of similar genes and know how are these things acting. And we're also able to look at the transcriptomic regulation and say, what are the pathways that are actually shifting? There's a narrow question around MOA, which is what is the target that this drug is actually binding to? And that is one that really helps to accelerate drug discovery downstream, but it is not necessarily something that we have to have early on, and it's also not something that's absolutely required. And so the exact binding target is something that we try to predict through Matchmaker, and we also test things that are showing strong similarity in the map to see if we do see that binding. And it is something that is an iterative effort that we particularly pay attention to in later stages of drug design.
Marissa, if you can stay online and answer some of the questions in the chat after this, that will be great.
Absolutely.
We can move to our next speaker. Thank you again, Marissa.